Moving onward.
Onward and upward.
All right, we have somebody, Kyle, aka Dawson, the IT guy from Tennessee, wondering if you have a place on Nautilus at some point. So it seems like you have a background in IT, but you are also interested in the biological sciences. I would say absolutely. There are lots and lots of seats available on EV Nautilus. You do not have to be strictly a science, biology, ocean person.
I mean, I think even like in my job with video, like
Yeah.
if you have a IT background with any kind of broadcast, I don't know, wiring and work like that, you could probably apply for the video engineer intern gig, as well as maybe work with like Mike and the data.
Yeah. Yeah, the opportunities on Nautilus
They have
are
IT
quite
experience.
diverse.
Yeah.
Join us, Kyle.
I was going to say, we have data engineers. We have, yeah.
Yeah. Shout out Kyle.
Do you want to look at this other colony off to the right?
No, I think it's the same as the other one.
Okay.
I wasn't even on SPL. Nice. I need to retire.
What?
Jeez.
Jeez.
It's kind of early, huh?
Yeah, no more working for me.
I'm done.
Calling it.
It was good.
It's a good run.
It was a good run.
Honestly, like, finishing pretty strong, to be honest.
We got something. Do we have something floating over there to the right?
Just kind of like a little fish or
Yeah,
something.
there is.
Oh, yeah, there is a little floaty fish
Yeah.
on the right,
floater.
channel 1.
Yeah?
Yeah.
Boop, kind of in the
Something
distance.
swimming.
Come on. Something
Let's
swimming,
go.
something swimming. We got a floater.
I hope it's a halosaur.
I do too.
I miss
It's been a
those.
while.
I do miss
For
them.
sure.
If it is, we finally have a winner.
Well, not really, because that
was
was the first
yesterday.
fish.
You guys see where it
Oh,
went?
yeah,
Sorry,
sure.
I took
So
my
there's
eyes
no
off
To
of
the
it.
bottom
Wait,
center.
Yeah, it's towards the bottom.
It's like almost by the porch.
I thought we only saw eels.
It's pretty low down there.
But there, yeah.
Oh. I lost sight of it.
Yeah.
Yeah, it's below somewhere.
Lost visual.
I think it went swimming.
Yeah, I don't see it anymore.
Yeah, it's left. Okay, that's
Sorry, gang.
That's okay.
All good.
that's
All
what
right.
it is. Fish doesn't
Yeah,
want to
no-
be
no biggie.
seen.
You can't control nature.
They've been running away from us this whole time.
Yeah, they've been quite shy.
Right. That baby fish, poor baby fish.
Okay,
Looks
another.
like our ridge is starting to turn on us.
Yeah, I called. I stopped our ship change because I had a feeling we needed to start heading more farther north.
It looks like it's heading further south.
The
The
ridge
ridge?
itself?
Yeah.
That's
Or
we'd be
we're-
going back down.
We're heading down.
So
So I think that waypoint 12 would be like
Okay.
northwest. Maybe like a 320, 320 bearing, but
All right, well, let me get back over.
ADA is still going to be swinging at 270,
Yeah.
so I was just going to let you kind of
Okay.
give me eyes on where we
Yeah.
need to go.
My apologies.
Ah, none needed. The other day when we were navigating the crevasse, I had no idea up and down.
There we go.
Yeah, Megan got a little irritated. She's like, when I have to annotate this, you're driving around in circles. It's going to make it so hard.
I actually felt that I was like, wow, whoever has to kind of like, I was watching like all the little configures on NavG and I was like, I still have very little confidence on where we went and who we are. And
Who we are.
I didn't know.
Not even where, but who.
No, I lost myself.
It's going to be so difficult to annotate.
I sympathize.
That's deep.
Deep.
A few minutes ago, somebody said, Megalodon watch for life. I'll miss you guys as a viewer. You've been keeping me company in the background a lot this expedition. I'm glad I've been able to see some neat animals and geology and learn more about the deep sea with you. Heart.
That's real.
Nice.
Thank you
Thanks
for
for spending
being
Shout out
time
you.
with us.
Shout
That
out
means a
whoever
lot.
that is. Yep.
Oh, we'll miss you all too.
We appreciate you all. for
I
tuning
know.
in.
One of my hobbies when I'm at home is to troll the chat and just call out everything the pilot does well.
Goodness gracious.
That's evil. I know the SCF is like, who is this? And how do they know what's...
Did they block you?
Yeah.
I have a feeling it's coming.
Yeah, one
The
more.
dark evil.
So we do have viewers all over the world, it seems. I'm seeing we have some viewers, of course, from UK, Germany, Canada, the US, Portugal, Poland, Mexico, Japan, Italy, Israel, France, Denmark, China, Brazil, and Belgium. And that's just a few of them that are joining. So everybody that's tuning in, thank you very, very much.
Shout out everybody.
Shout out everybody across the globe. Again, this is the 4 to 8 watch. We have...
Who is that floater? It
I was
moves
gonna
at a
say.
different speed as the other particles.
Yeah, just under an hour left of this
Zoom
watch
in.
before we switch on over. And once again, this is our last dive of NA174. Is that a comb jelly?
Yes, it is.
What?
Look at that.
Oh.
Tina 4.
I was like, that is not a particle.
mesmerizing.
It's
Oh,
a
I
floater.
love it. It's so
Wow.
hypnotic.
Wow.
Beautiful.
Look at the yellow coloration. Whoa, whoa.
Come back.
I know. See if we can find it again.
What is, wow.
So
It's
cool.
like electro waves.
It's coming at us.
Yeah.
It's coming right.
Hey, come closer.
Come
Come
on the porch.
closer.
Come on the porch.
Oh my
Whoa,
gosh.
it's like in the shadows. This is so close.
It is coming with us, clearly.
That was so amazing. That was great, Fanny.
Yeah.
Do you have any clarity on where the ridge is?
That head?
All right.
Yep.
310.
Can I get a adjustment on the focus, please?
I am trying. Actually, no, like I'm trying and it's not. So hold on a second.
Okay.
Yeah, it's focused on the particles right now.
Yeah.
Yeah.
I love how rainbowy the
That's what I'm
comb
saying.
jellies
Yeah.
look like, but they're, what is it called? Cilia? Scylla?
Yeah. Is there any
I
way
think
to reset that or restart it?
I wonder if the-
Amazing.
And aren't those like crystal-like? It's not, or I forget. They're not like.
They reflect
You
the
don't have any-
light.
Yeah.
That's the way they
You have iris
reflect the
control?
light.
Yeah, so they're not emitting light
Yeah, I have iris
themselves,
control,
Yeah,
but I
but
don't
it's
but
have
they're
reflecting
reflecting
it.
light as it's like teeny tiny, I don't want to say tentacles, but maybe that's the word for it.
No, I think it's series or something like this.
Yeah.
Mike,
There it is.
there we go.
Okay. Thank you. I don't know what you pressed, Danny, but thank you.
I just tilted up.
Oh my gosh.
I was about to say we could restart the camera, but
Oh, thank you. Yeah, cilias, that's what it is.
That was cool.
Amazing. Yeah, so once again, the comb jelly that we just saw, the really amazing like rainbow effect that you can see is light reflecting off of the cilia of the comb jelly. So it's not coming from the jelly itself. That was neat. I definitely wasn't expecting to see one of those on our dives.
Expect the unexpected.
I guess so.
Looks like we're heading down.
Yeah, that ridge like goes down and then it should come back up.
Okay.
Our porch has been popping today.
What has?
Party. Our porch
The porch has been popping.
has been popping today.
Hopefully it doesn't pop too much.
It seems pretty normal.
Don't pop
I don't
off.
think you guys know what I mean by popping.
Like it's popular. Yeah. But then I took a different route.
Yeah, I was like,
yeah. I know what you meant. I just shifted gears a little there.
There's still some trauma associated with the porch.
Yeah. That's what I was not trying to get to.
No, we're past it. It's okay.
That current
We're
looks
in a
pretty
better
strong.
place now.
Yeah, a little bit on the strong side, but I'm also going downhill, so having to watch the BSR.
Sunrise still looks beautiful.
Are you looking at the wire or where are you looking, Belle? The sunrise is kind of risen, right?
It's coffee and cigarettes time for the men back home.
Yeah, definitely stronger current here. Come down on Atalanta.
I'm still only 19 meters off the bottom.
Yep.
Someone said, we will certainly miss watching and listening to y'all. Your banter makes watching and learning even better. Blessings to each of you as you continue on with your life journeys.
Thank you.
Thank you
That
very
is so
much.
sweet.
I love that.
I'm over here trying not to cry, reading these messages in
No.
the chat.
Everyone is being very, very kind.
Very kind.
Thanks for sharing all of them, Brittany.
Yeah, I'm trying, especially because it's our last watch. I definitely want to acknowledge
They're
the
still making moves.
We're
kind
still
comments
making
coming in.
moves, bearing 3110. Or 310.
The chat has definitely been making this whole exploration very memorable.
Yeah.
I really do
Yeah.
feel like there's like a 9th crew member in here, you know? Well, 10th. We have a net in and out. And Annette is now here. But like the chat feels like a 10th crew member, you know, that just kind
I
of hangs out
agree.
and puts their input in.
That's the goal. We want everybody to feel included in this exploration.
Yeah.
It kind of is going to slope down.
Looks like we
Or
did a
goes
good
up.
job at that.
I hope so. Yeah.
Horrible light, but I think the wind is picking up a little bit, so maybe the ship is...
Okay.
I'm in denial, guys.
No.
Another one. Going to miss you all. You have all made the days fun and interesting. Truly love you all and brightest blessings on your next chapter.
We love you too.
Aw,
Yeah.
so
Yeah.
kind.
Thank you.
None of this would be what it is without the entire international viewer.
Oh yeah, for
Oh,
sure.
yeah. Easily. We're all
Without
explorers
viewers
here.
like you.
Somebody was watching us as their Achilles tendon was healing.
Ooh.
Yikes.
Oh,
I'm glad we could keep you company.
A lot of
Speedy
people who are
recovery.
healing and coming here to watch things.
Yeah, I mean, I have heard like it's just like so mellow, you know, it's just nice thing to kind of watch most of the time.
Mellow us 8 hours ago. Second grade classroom.
Yeah, except for whatever was happening yesterday. I don't know what
I
that
don't know.
was. We have no explanation for that one. You have nothing to say to
Very
ourselves.
chaotic.
I don't know who that was.
We were different people
Benny
then.
tearing up. Benny was tearing up, so that's how chaotic it was.
I think it was.
We were different people yesterday.
Such a highlight. I will never forget that. Benny tearing up, Hayden getting cramps. Did you just? Oh my god.
By the way, people listening, Fanny was tearing up with laughter. It was nothing. She was laughing
that
too
was
hard back
so
here.
funny.
Yeah,
It's just that
we
little-
lost focus for a minute.
Yeah.
a good minute.
Maybe we missed the megalodon because
We
of
weren't-
that. Can
we
you
weren't.
imagine?
We weren't locked in.
Mike never took his eyes off the
I
screen.
would definitely say that was
We're
But was he
fine.
busy with the winch TV though?
Oh.
Range TV.
so many highlights over here.
You're going to miss Winch TV. I know.
I'm never going to be able to see the
I'm just
winch
gonna have
the
to-
same again.
Oh.
I'll just put it on all the TVs in the lounge till we get back.
All those unsung heroes in the winch, the 100 mics.
The 100 mics.
The 100 mics.
Reminds me of 100 gags. Like, that's what it sounds like.
Oh my god.
100 mics.
These
Watch them all
sponges,
drop at once.
these sponges remind me of the Jetsons little sofas. Do you guys remember those?
What'd you say?
The Jetsons, like the TV show. They had those like little round seats, like so spacey. These sponges remind me of them.
Oh man, I don't know. That's a deep cut. I don't know.
You don't know the Jetsons?
Yeah,
Okay,
that's very,
Sorry.
like the couch in the Jetsons is very specific.
I don't know. I guess I just always saw it and thought it was so comfy. Like, it's my dream to have a Jetsons couch.
Huh.
I feel like you might be spot on with that. Like, I feel like if you were able to, like, artificially replicate the
a little
sponge, like,
fish.
structure.
You see a fish?
Yep, right. It's almost center screen.
Center
if you can
screen.
zoom in.
I would be happy to.
Good eye, Mike.
I don't see it yet.
It's
There
small.
it
I
is.
see it.
I see a
Good
fish.
one.
I'm trying.
Oh no.
It really blends in.
No.
with the
It
rock-
does.
Rockscape.
Is it similar to the one we saw before?
It looked like
I'd
the
say
one we
so.
saw in the on the rock.
Yeah.
It's
It
like
looks like it's
swimming
swimming
away.
in reverse.
It's, it's like trying to swim against the current.
Yeah.
Baby rattail.
Come this way. Come join the porch.
Hop on the porch.
I'm getting my belly.
We'll take you where you want to go.
Looks like a little cascale or something.
Cascale, you think?
Good flying, Mike.
Yeah, that was awesome.
All right, go ahead and come wide.
I didn't even see that.
They're like the exact same color as the rocks.
Yeah.
Literally. Look at that. Just blends
Hey,
right
It
it is
in.
Really
disappears.
small.
It's that time to
Okay.
switch the pumps. So,
Pump time.
okay, turning off 12.
14 is what we're starting up.
Yes, 14.
End of 12, start of 14, and that's 266.
Correct.
And the depth?
2480.
Okay.
This breezing G is back.
I was about to say.
It's been a while.
Yeah.
We
can
were
say
seeing
that
so
now.
many in the beginning.
There was so many in the beginning.
All right, you want the volume?
Give me 1 second.
ROVs are fighting.
All right, give me that volume.
152238.
And then can I get time as well? The time ran?
54 minutes and 21 seconds.
Okay.
Thank you.
Perfect. Thank
Yep.
you.
And your ship's still on the move?
Still stepping 310.
Roger.
And it should be happening an okay time, considering the vectors are with our movement.
Okay. Sounds good.
Just think the ROVs are catching.
Yeah.
Like sails in the wind.
Come sail away with me. Sorry, bad, I know.
It's okay, I forgive. But I don't forget.
Nor would I expect you to.
New rockscape unlocked.
Rocks, rocks, rocks.
Getting some more kind comments. I can read a couple right now. Love this and that you're all streaming this. It really helped demystify the experience and share it with a wider audience. You're amazing for doing this, and I hope we see more live streams like this take off. Thank you. I'm
That
so
means
glad
a
to hear
lot.
that. Yeah, I'm glad that we were able to help demystify the experience. That's a huge deal.
Yeah, give
Yeah.
it more of that human feel.
Well,
I
and just
guess there's something
Shouldn't
on
say
that
that.
rock here.
You want to
There was
come and
something
take a look at
floating,
that?
but
Yeah,
all
let's have a quick look at that.
Sure.
shout out to the chat. They're just making me feel all type of
I
ways
know.
right
Yeah,
now.
I feel like
Keone, don't cry.
it's all right. Tears are healthy. Let it out.
Brittany's like, cry.
Cry.
On shift.
Either way.
It's definitely been a surreal experience
Yeah.
for sure.
No, I think it's just a sponge stock. We can continue.
Okay.
Just wanted to check it out.
No, no worries.
Someone is wondering, what are our plans after Marshall Islands? Does anybody have anything happening afterwards?
Sleep.
College.
Sleep, college,
More conservation work for me.
conservation work.
More filming over here.
Woohoo!
Mapping crews,
Woohoo!
maintenance.
Yeah, a couple more cruises for me, and when I'm home, I want to rest, reset, eat nice food, hang out with all my lovely people in my community. And it's the end of the summer, so
Sounds awesome.
I want to camp.
You get some good quality family time,
Yep.
and then I'm off to go and do a mooring cruise.
Mooring cruise.
Moorings.
When I finally get home.
How long is that one going to be?
3 weeks.
Okay.
Yeah.
When I finally get home, I'm going to Disneyland.
Disneyland.
You went to Disneyland?
Never been.
Awesome.
You've
Me
never
neither.
been, Danny?
Huh?
I live not too far from Disneyland. You should hit me up. If you
Oh,
want.
really? Do you go often?
Oh no.
Oh.
I don't live far from it.
I might be going in that area in January.
Oh my gosh, everybody.
I think
Hit me
the
up.
first week of November is when I'm going to go to
Costa Rica.
Disneyland.
Okay.
I wanted to go for Halloween, but I don't have a cat babysitter during the month of
Oh.
October.
I wish I could help. I would happily watch your really cute cats.
Megan's got another cruise, and I take care. My partner and I take care of her cats while she's at sea.
I like how you guys do that for each other.
That's nice.
It's a good trade.
Are your
I've-
cats all friends? Or do you just go to her house?
We just go to her house. I check on them once every other day.
Cute.
Make sure their food and water is good. Clean the toilets.
That makes sense. I'm sorry, Brittany, I cut you off.
That's fine. I was just gonna say, after I get off the ship, I'm doing a little detour. I'm gonna go spend some time in Australia and New Zealand.
Oh, wow.
I can't wait to hear about it.
Yeah,
Hopefully they
super
give you the
excited.
haka.
What's in Australia?
Hopefully you
The
experience
Great
that.
Barrier Reef.
Nice.
Yeah, so
We're at in New Zealand.
Does it say
Christchurch.
kangaroos?
Christchurch.
My favorite city in the whole world.
Really?
Do you need recommendations?
Sure.
Did Brittany
I'll
say
take 'em.
Australia or New
Food?
Zealand?
Both.
I'm doing both.
Oh, okay, thank goodness.
Oh
Oh my goodness. I actually feel awake is the worst part.
That was great. Yeah, I'm doing both.
I'm gonna
Did I
cry.
not tell you about my tattoo, Bella? The meaning behind my,
Sail away?
yeah, that whole thing.
Oh, yes, I remember.
Yeah. So,
Remind me, what city is it that you're
um,
kind of
so
bucket checklist stuff?
it's actually the Great Barrier Reef itself, because she
Oh.
mentions the coral
Ooh.
sea. What?
So, if you get the chance in
Oh, I thought you saw
the-
something.
I know,
was
me
like,
too. I was like,
No.
"Whoa."
The whole room
Sorry.
went silent.
if you get the chance in New Zealand, they have one of the only colossal squid remains ever
Oh,
on
really?
display. I don't know what it's in the.
There's a different
Just in the
coral
whole
here.
country.
Yeah, there's
Coming back
something
to
on
it?
the
Oh,
left.
fine.
Yep.
Is it like preserved and all that?
Yeah.
Wow.
And
Museum of New
shrimps.
Zealand.
And the shrimp, I think, swimming shrimp.
Te no te pua.
Hello.
Yo.
That sounds really cool.
I can't imagine.
Take pictures for us, Brittany, if you do go.
Mhm.
Oh,
Yeah,
yeah.
that's going to be an amazing trip.
Oh,
I do plan on
Thank
going
you,
to the
everybody.
California Science Center when I'm in LA. though.
Danny, you have to come through.
Definitely.
I was thinking that same thing. I don't know if I'm ever when it'll be for me, but I'm like, wow, I gotta go.
Oh, I'm for sure coming when
I
I
hope
go
you're
in
still.
January.
I don't know when it'll
Reunion
be,
at the
but
Science Center.
I hope you're still there.
Coming, I'm coming.
I'll text you when I'm there. I'll be like,
Okay.
where are you?
Sounds great.
My luck. I'm not working
I
today.
know. The one day I'm not working.
You're going to walk in and say, I know somebody.
All right, well, just trying to land. Okay, video.
Okie doke.
I know I might call you video. Kelsey, zoom in, please.
You got it.
It's just easier.
I don't mind. I don't get
Got other things to focus on than trying to remember people's names.
That's
Besides, we have
another
nav and we have video.
crinoid. It doesn't look super healthy. Some damages here.
Swear I didn't touch it.
I was gonna say, Mike, what did you do?
What have you done?
I didn't touch it
You looked at it wrong.
yet.
You gave it a look.
A wide-eyed stare.
The wide-eyed Mike stare.
Don't do that.
Yeah, that coral somehow doesn't look at its best.
I can feel it.
Ooh, I can see it.
All right, zooming out. Thank you.
That's
great.
Come on out.
Hold it together, guys. We got 25 more minutes.
25 minutes, no. Someone's wondering what they should add to their ocean tattoo. Everything.
I really like Kelsey's Halazor outline tattoo that
Alright.
she's gonna do.
Come on up.
I'm sorry, Keone, I didn't hear what you said.
We're just talking about tattoos, and Keone was saying your halosaur tattoo is going to be great.
Oh, thanks. I'm so excited for it. I think. I hope we can find
that
time
you?
for that.
Huh?
There's so much to
I
do.
don't know. I couldn't tell if somebody...
Iridogorgia would be a cool tattoo.
I'm just over here throwing out random numbers.
That'd be a crazy arm sleeve.
That would
Right?
be very
That's what I'm
evil.
saying.
2, 4, 7, 9,
No, I'm...
6.
You should get the winch.
The winch.
Winch.
The winch tatted.
On a TV.
Get the 100 mics on there too.
Yeah. 100 horse mics.
Oh my god.
And Katharina watching with her dog Lucy. Thank you so much for your kindness, your tuning in and watching multiple times. I think I remember you said that you were on the mend, so we hope that you're feeling better and better.
Shout out Katharina.
Yeah.
Hi, Lucy.
Give Lucy the extra pet.
I hope Lucy
Shout
just
out
heard their name on the
Lucy.
screen. They're coming from the TV.
Started
It's like,
barking.
huh?
Someone said yesterday when we were talking about Gabby, there's a cat that they have named Gabby, and it came to the screen.
No way.
Yeah.
I
is
love
so cute.
that.
Oh, those are beautiful. There's a nice
Wow.
sponge
You've
and a
collected
coral
a
on the left.
sponge.
Wow.
It's probably a shrimp in there.
I think so, something.
I like the top view. That's cool.
Sounds really
Yeah, it is
cozy
really
to be
cool.
a shrimp and a sponge like that.
Looks like a web.
Yeah. And if
Alright,
we zoom
video,
in on
you
that,
zoom in.
we might be able to see inside a little shrimp.
That'd be a cool
Hold
shot.
there.
This is a really cool angle that we're taking a look at it.
Mm-hmm.
Quick scope.
Okay, you can keep zooming.
Look how slow.
It's like a spider web almost.
Is there any associates within?
Knock knock. Anybody home?
I don't
Don't
see
see
anybody.
anything obvious right now.
Zooming
Yeah.
some more. It really does look like a spider web.
Just a touch. Thank you.
That's the tattoo I want, right there.
There you go.
There you go. That's so cool.
Solid.
Yeah, I don't see anything right now.
Very cool webbing.
It's still a super cool view.
Mm-hmm.
I'll go tell this tattoo artist, I want a sponge tattoo. I'll end up with like a SpongeBob. Or dish
With the
sponge.
spider web.
Okay, go ahead and come wide.
What?
All right.
What? That's not what I wanted.
Can I do a little DVR reset?
Yep.
Colophacus, sponge.
And keep going, or
Yeah, keep going.
okay.
We have someone wondering, how often do we see nautilus? And I believe we've seen it once. So Ocean Exploration Trust has been operating since 2008, and I believe that the very first sighting of a nautilus from EV Nautilus was last year.
Wow.
Delta's 15.
That'd be a cool tattoo.
A nautilus, yeah. Those are
Marley
just
has one, doesn't she?
amazing. I don't know. I wouldn't be surprised. It's a pretty big bursingid on the top right coming up.
Coming up,
Wow.
yeah.
Growing back some arms.
Lost a few arms.
Wow. The other ones look long and healthy though.
Yeah. And there are a few arms just growing back.
Someone got hungry.
Go ahead and keep moving forward.
Keep moving forward.
Okay, should be cresting over this. And moving forward.
Wow.
I'm switching classes. Everybody take a lap from the classroom. That's sort of a whole year of aeronautics, right? We can act like that's a casual thing.
They're just like,
Actually, I meant like a TikTok picture or something.
13-meter delta.
I'm wondering if maybe once we hit the 10-minute mark or something, like 7:50 or so, we can say our like formal goodbyes to the chat.
Oh man.
It's only
Sounds
right.
too sad.
There's a fish, by the way.
Sad, but it's only right.
Channel 2, right in the middle there. So Atalanta, Cam,
Oh.
you can see a
Yeah.
fish there. Sorry, should have been more specific. But yeah, just throwing it out there in case anybody wanted to
Yeah,
say
That's a
farewell.
good idea.
I'm down.
might as well, right?
Yeah.
Looks like the ridgeline is starting to move north, but then also heading back to the west again.
We got 5 minutes. And the question is, will something crazy happen in those 5 minutes?
Megalodon!
So again, this is going to be the end of the 4 to 8 crew. Coming up in just about 15 minutes, but there is still plenty of dive left.
350.
Oh, yeah.
Oh, 330.
I'd say stick at 310.
Okay.
So it looks like it's starting to come around.
We've got a bunch of ship movement kind of in the bank, so I'm gonna let Ada keep swinging with it.
Okay. Wow, it looks like it curves up here.
This is cool.
We were in sync right there.
What do you mean?
Both of us
We
very
both
curious.
looked at- yeah,
We
we're
just
curious
turned
what's
around at
happening.
exactly the same time.
Turned back at exactly the same time.
That's funny.
We spent too much time sitting next to one another.
We are.
We're all polyps on the same coral
We become polyps and
Oh yeah.
at this point.
It's a wonderful way to put it.
I really
I love it. I love it.
I don't share my food though. Polyps do, but I don't.
Whoa.
Sharing is caring, Fanny.
Fanny will not share the Nutella.
Well, the Nutella is long gone, no?
Yeah, I
Oh,
think so.
man.
I don't know.
You're gonna have to
Yeah.
get yourself some more moonlight cookies before you go home.
I know. These were good.
Fanny, are you an only child?
No, but with my
Goodness.
brother, we were constantly fighting for food. It's like, you know,
Are you the younger sibling?
I grew up like competing for food. That was crazy.
That was
With my
crazy.
brother, yeah, we had to hide food. It's like you had to eat fast, otherwise the other one would eat it.
Are you
I have
you
a
the
younger
youngest?
brother.
Oh, wow, you're the older.
Yeah, 3 years difference. But yeah, life was competition.
Wow.
Life was
Tough
competition.
times, tough times.
He's not a deep sea biologist, right?
he's
Come
not.
on. So,
What does he do?
He is like he works in software development, so he works for a startup back in my hometown, like developing apps and software programs.
Oh, very
Nice.
cool. So two very
Looks
smart
like this is going to be our new heading for a little bit as well.
people.
all right, and you're at 370?
270.
27. I in my head I was saying 2.
I mean, she's just making up headings over here.
I was going to say, that's a new degree.
That would just be 10. I don't know.
You're doing fantastic. What are you talking about?
370, please.
Yeah, really. Actually, I'd love to see what the bridge says if you tell them that.
I would not show up to breakfast. I would not make eye contact with work.
Oh, someone said you guys are my favorite crew. You got to lock the door.
No.
Done.
It's
If
broken.
only.
We don't know what happened.
If only.
Uh-oh.
It's stuck.
The
And
Nautilus podcast.
Someone's wondering, what are our favorite critters from this expedition?
Halosaur by far.
Pancake fish.
Pancake fish was
So
cool.
darn awesome for me.
Yeah.
Definitely a highlight.
Yeah, I also agree with the pancake fish.
Yeah,
that
that
was
was-
so cute.
Really, really cute.
I gotta say, the crinoid hats were pretty cool too.
Oh, the fashionistas.
The fashionistas.
Yeah.
The megalodon we most definitely saw.
Yeah, we definitely saw
Very
Megalodon.
low.
No big deal.
Very low key.
It was just in the background.
Mm-hmm.
We didn't want to make a big deal out of it.
It was
no
just so
big
big it
deal.
made the sat
Major.
feed cut out for a second.
What was that, Kelsey?
It was just so big it made like our feed cut out
Oh,
for a
yeah,
second.
yeah, yeah,
Yeah.
yeah,
Yeah,
yeah, yeah,
no one
yeah,
saw
yeah.
it.
Yeah, yeah, yeah.
No big deal.
It's fine.
Just another day on the Nautilus.
Mm-hmm.
Nothing major.
Just another day.
All right, shall we begin our...
It is about
Our
that
big
time.
goodbye.
I hate it. I don't want too.
All
But
right.
yeah, it's getting to be about that time. So
Hold up, what's that? Is that a sand patch off to the left? Yeah.
Yes.
Okay.
We
Sorry,
got
it was
another
squid shaped and it
Hayden.
scared me.
There's
Sorry.
another
Cucumber.
transparent cucumber here with
Oh, yeah.
showing us its guts.
showing us its guts. The way you say it. Showing us its guts. Okay. All right. Okay. Bye, everybody. Thank you so much for watching. It's been a pleasure hanging out with each and every single one of you. Thank you for tuning in, especially those of you who are like here for every single dive. That's very impressive. It means so much to us. We will miss you so much, and we hope that you continue exploring, even though this expedition, this shift, this watch is no longer going to be here. We'll always be together in spirit. Thanks so much again, and take care.
Kelsey, could you zoom in, please?
Yep.
All right, is it my turn?
It is.
Well, hi everyone, and thank you so much for following us during this entire expedition. And I think I talk for everyone saying that it has been really a pleasure working with this crew
Yeah.
on this watch. Really enjoyed it. And my last Nautilus expedition was 10 years ago, so I'm really happy that I was back, and I really enjoyed every single minute of this expedition. And hopefully we'll be back soon.
Yep.
Thank you.
Hi everyone, Keone here. Oh man, where do I even start?
I know.
It's been a huge pleasure exploring
Okay. Go ahead,
my
come
own
wide.
backyard with you guys and having all the loyal viewers just making every single moment of it nothing but a great time and just great memories all around. It's been real for sure. And hopefully we all get back together sometime and start our own podcast. The chaotic crew. Yeah, but much love and blessings to all of you guys,
Yeah.
and thank you again.
Your turn, Hayden.
Okay, it's me now.
Yep.
Hey, you guys. I just wanna say thanks for like tuning in, and thanks for joining. And I really don't know what to say. Like everything that we've done, everything that we've seen it's just been fantastic. And I hope to continue. I hope that eventually I'll be back. I really do. And I hope that you all continue to explore as well, because this is important. It is, yeah.
Chase them dreams.
Protecting these areas, understanding these areas, it's really important. And one word, I haven't came from an area where this typically happens. I'm very fortunate to be able to do this from where I come from. And so no matter where you are, no matter what you're doing, if you want to do something, you can do it.
You
just
can
gotta,
do
just gotta
it.
have a lot of spirit.
Thank
a
you,
lot of hard work.
Hayden. Your mom is
See you
very
guys.
proud of you.
Shout out
Quickly,
Hayden's
No
Bella.
way.
mom.
Quickly, Bella.
Okay, I gotta go quick. But
Yeah.
I just want to say, like, I resonate with everyone's statements, and I just want to, like, amplify that. I feel the same way, and just want to throw out some gratitude for, like, the Marshallese and the Solomon Islands, and getting to take up space and explore these waters. It's been a real honor to be a guest here, and that's really what I gotta say. Thank you, thank you, thank you.
Very good. Anything, pilots, before you swap out? I know you.
I'll be short and sweet. It's been a pleasure piloting with you guys. It's been my first time as a Hercules pilot for Watch Lead, and it's been a wonderful time. And I'll see you guys on the next season.
And I'll go real quick as well. Yep, it was great sailing with everyone here, from people at home to the people here in the van and on the ship. I really appreciate everybody tuning in and getting to talk story, and yeah, it was fantastic. And I'm sure I will see you guys on future cruises, and I'll drop the mic there.
Yes.
I guess I'm going to close it out. Thanks, you guys, for everything. You've been a part of us, like, living what I would say is our dream. So thanks for being along with it. And yeah, keep sticking on the stream. We still have 5 more hours. I know it sounds like we're ending, but we're just swapping watches. And yeah, thanks for being so supportive and helping us live our dreams and get science out to the world and communicating the importance of it. And thank you so much to the people from the Solomon Islands and the Marshall Islands for hosting us.
All right.
And with that, 4 to 8 crew for NA174.
Signing out.
Over and out. I see we still have some more comments coming in the chat. I'll be sure to share that, share these comments with the rest of the crew, but we do need to switch it over. And so it's now the 8 to 12s coming on in. Still got hours of exploration. Once again, thank you everybody and take care.
Take care, everyone.
Bye,
Much love.
everyone.
Thank you, you guys. It's been wonderful.
Two 270. We don't care about those, right?
Oh yeah, we already saw a
Dime a
bunch of
dozen.
those.
Dime a dozen.
There's like so many during breakfast.
Yeah, right.
And one bowl of soma here, a bowl of soma there. Find me
Carrying
a
on. Fish.
yeah, find me a fish or an octopus.
Not a lot of fish either. We've seen only a few.
Yeah. We saw a few last night.
Wow, there's a lot of drops of water
Yeah.
in there now.
So the terrain is kind of like going up
That's
and
cool. Let's
down.
look at
Oh, that one we haven't seen yet.
Gonna give you a little zoom.
Look at those tube feet.
Yeah.
It looks... Oh, my goodness. Man, we're not even on my right page yet. Well, it looks like Astenactus. It's very skinny. The skinniest Astenactus I've ever seen.
So yeah, we've been following generally the waypoints, but that ridge is relatively narrow, so we've been trying to stay on the ridge, and it kind of like goes up and down.
Yeah. And geology does what it wants.
Right.
There we go. Zoom out.
Is that for me?
It's time for Levi's to wake up.
He's got his alarm just goes off and off and off.
Oh, is that the last one? Okay, because if you're not up by now, you're late.
Yeah. Knocking on your door.
And we're like, where are ya?
I hate that.
Yeah, we're good on this sea star. Thank you.
Right. So you know this is a stick lock button too.
Oh, that's a button?
Stick.
It just a boop. I didn't know about that.
Yeah. I added that.
What a feature.
Recently. But I never use it. I don't know if anybody uses it, but
I didn't.
it's there.
I didn't use it because I didn't even know it was there.
This is too awkward, you know?
Yeah, the poke
I
the
think
button.
it needs a button over here, actually.
Yeah.
That would be better.
We need more buttons.
Yeah.
Always.
Never enough.
Except the button.
Except for button. We don't need the button. The button to rule all buttons. It allows you to push the buttons.
Yeah. What it does. If you take the button off, then you can't. None of these. You can push them, but they don't do anything.
To activate the buttons, you must include the button.
Which is the same thing up here.
Yeah.
This ridiculousness.
Why do we need that?
Back up to the button.
If the green light is always on, does it indicate anything?
Well, if it's not on, then these don't work.
Then you can't activate the other buttons.
I don't know why you even
It's the
have a
foolproof
button
button.
that makes it so other things don't work.
I don't know.
It's a feature.
It's a feature.
Yeah. Child safety.
Yeah, I guess maybe we just need some child safety logs.
Yeah. That's what it is. Yes.
Yeah.
I mean, we have the protect button and the lock button on your guys' panels.
Oh, you do?
Yeah. On the bottom.
So you have
under the purple.
Too.
Oh yeah. And if you push that, well, don't do it now, obviously. But if you push that, then the whole panel's locked.
See, that makes sense.
If you lock the whole thing out.
Just lock it all out.
Yeah, that's more if just like people are mashing things, or I don't know, you're moving it or something.
Right.
I feel like we keep heading.
I am much better today.
Good. I'm glad you're feeling better.
I don't know why I go from.
I don't know. That's a nice big lolosoma. Usually there's like shrimps inside there.
We saw some with the Tinophore, and I think, is that like a black coral next to it?
It looks like a heteropathy.
Yeah, it looks like it. We've seen a few also.
Man, look at all those water blobs.
I don't like that.
No. I'm going to tell the cheese about that.
So would you consider this one to still be healthy?
Yeah, this one's still healthy.
Megan, do you know by any chance why they're yellow?
They have like a chemical in their tissues that causes yellow color. I believe it helps avoid predation, but I'm not exactly sure.
Yeah, we were wondering about that earlier.
Some sponges are just yellow. I feel like more sponges in the Atlantic are this yellow color. But there's a few of them here that have that color too. Let's look at this black coral down here.
Yeah, it's like some right over there. There it
Do
is.
I need to relocate?
No.
It's just here. It's very small.
Come on, I know I have more, Pan.
Move over, moving over.
All right, so this is the last dive of the expedition, which means this is the last 8 to 12 watch for this team. I'm sitting in for Giselle for a little bit in the back row. This is Marley. Do you guys want to do some quick introductions since it's the last watch, or how you feeling?
Lining up on the wrong thing. Is it
Sure.
okay?
Yeah.
Let me know when you're ready.
Somebody going?
I was just zooming.
Yeah, I
good?
think we can zoom out. Thank you.
Wide.
Oh man, look at all those water drops.
I know. I wish we could like...
And I can go next if we do introduction. Hi, I'm Fanny. I'm actually not on this watch normally. I'm on the 428, and I am just temporarily here while Val is finishing a live interaction. But I am one of the co-lead scientists on this expedition. I am a deep sea biologist, and I'm based at the University of Hawaii at Manoa, where I'm an assistant professor.
Hi, my name is Luisa Dueñas. I am from the National University of Colombia. I'm based in Bogota, and I'm also here with Ocean Census, and I'm seated in the Science 2 seat. I'm helping with all the biological samples we're collecting in the expedition.
Good morning, ladies and gentlemen. My name is Jovel Warnendanko, attending the University of the South Pacific from the Solomon Islands. I'm an ocean science intern serving as a data logger.
Awesome. Thank you, back row.
In your front row, I'm Megan Putz. I am the navigator on this watch. I am from the University of Hawaii, and I do the video annotation from these dives.
I'm Robert Waters. I'm in the Herc seat, currently going downhill, which is the wrong direction.
Well, I guess we're going to have to go downhill to go uphill. We were pointed like down the seamount, but it was going up.
Yeah, that ridge keeps doing that.
It's bumpy.
Oh, hi, I'm Levi. I am on the Atalanta seat. I'm a student at Columbia University, and I'm also the ROV engineering intern.
Hi, I'm Amber Flynn, the video engineer. Yeah, and I, like Levi's, was originally a intern back in 2016, and it's been great to be invited back since. Get so many good buttons.
I'm
Annette Govindarajan was one of the science co-leads studying the distribution and diversity of animals in the deep sea using eDNA and our new eDNA sampler on Hercules.
Fish in Atalanta view.
Yeah.
Ah, it's gone.
I think it was the Bazazetus.
Are those water drops in that camera as well? What's happening there?
Yeah, it looks like on the bottom.
Have you seen that happen before, Robert?
Yeah, on the Zeus Plus, like, we have like one of the first models, and the seals are not ideal. So we need to send it out like almost every year to get it resealed.
So
this is the deepest dive of this trip, so maybe that has something to do with it. I don't know.
Bill, I'd like to turn off the pump 14 and start pump 15. Can we note that in the log?
All right.
Sure thing.
You got that? Okay, 14 going off. And what's the depth?
The depth is 2465.
I'm sorry, what was that?
2465.
Okay, thank you.
Is that a fish?
That is a fish.
Let's say hello.
I can give you the volume whenever you'd like.
Ready.
204050.
All right, thank you.
Thank
this
you.
is 267.
Yes.
Okay.
Has a
This
isopod.
is a Bazazetus.
Does it have some parasites on it?
It's a parasite.
Yeah.
It might be amphipods. It could be an isopod. I'm not sure.
It's blurry still.
You know, maybe we would stop moving its tail and swimming
Yeah,
away
can you
from
stop
us?
moving for a second? Thank you. We appreciate that. Show us your parasite.
Just a little passenger parasite.
See if I can get any more zoom on the parasites.
Yeah, he's it's getting away. We're at the end of the rope. This is quite downhill.
Well, sometimes you gotta go down to go up.
Robert, can you explain briefly why and how ROVs aren't really designed to go downhill?
Yeah, so it's Herc's just floating like straight, right? It's not like in a car where as you're going downhill, you're pointed downhill. So the ROV just sits level. So when you're going downhill, you're looking out into blue water because you can't look down, you know?
Yeah, the ROV
So
can't
the other way around,
pitch
if you're
itself
going
forward.
uphill, you're looking right into the wall. Well, we don't like downhill.
Yeah. But if we
So
point downhill, then we'll just keep
if you
going.
if you look at the rear camera, it's like dragging in the dirt. So the front camera is out in the blue water, and the back end's like dragging on the rocks. So. And people like to look at stuff.
Yeah. You know me.
This is true.
Is that why we're all here?
I guess some people like to look at the blue water.
Getting pulled.
Sorry. Sorry.
All good.
No, we're just getting reoriented.
Yeah, so let's start heading 315. That should help us go upslope eventually.
Right. Going down.
Going down.
Really going down.
Yeah. Oh, there's another fish.
Let's see which one this is. Oh.
We just got a question about what do fish down here eat? Doesn't seem like there would be much food. I don't know if, I don't think we have any fish biologists on the science team in the van right now. Does anybody know?
I like, oh, it's Aramicles. Oh, that's a good one. This one is a male.
It's pretty jittery.
Yeah, it's fast.
How can you tell that it's a male?
The pectoral fins, you see how long they are?
Yeah.
Yeah, the males have these long split pectoral fins.
This is a different fish.
Yeah, it's a different fish.
There's two fish.
Okay, I retract my earlier statement. Megan, what do these fish eat?
So they eat like small crustaceans, like amphipods and isopods and polychaete worms. They're scavengers down here in the deep, so if there's any sort of, food fall, they're going to go to that. Oh, that's a good view of those
That's a
fins.
good shot.
Isn't he, he's so handsome. Yeah, look at that.
Oh, this focus, sorry guys.
There was something on its tail, a caudal.
Yeah, there might be a parasite.
Yeah, if we get a view of that. There.
Oh yeah, sure enough. Good spot on that.
You can see it in the after action, maybe.
And an instant replay. Are we zoomed
It's
out?
right there. It's in your view somewhere.
Right there.
It's almost on the camera lens.
Where are you?
Right on the porch.
Come on, Megan.
Going down. Going down. Where?
Not enough beans on the down.
Yeah.
Oh man, it's under the camera.
Yeah. Like, I can't see you. Where
Camera
are you?
shy.
There we go.
There it is.
There are not enough beans on the boat either. We had the very sad news last night that we ran out of coffee.
Yeah.
The coffee for the front espresso maker. There's still the other coffee.
That's good news for you though, Val. You
See
won't
what
have
hands
people outside your door grinding away all day.
You know, that actually doesn't bother me because I love that sound.
No.
That means it's time to get up and make some coffee.
Yeah. I would be real upset if I didn't have an emergency supply of coffee. And we're also on our way back tomorrow, so it's fine.
Yeah. We'll make it
We got
work.
some good pictures of this one.
This is beautiful.
Yeah, it's hard to tell Eridmictes and Basazetus apart unless you can see those pectoral fins. So the Basazetus don't have that dual lobes of the pectorals, but they have the same sort of head shape. And so when you do see a male Eridmictes, it's really obvious, but the females look almost exactly like Basazetus. Whoop.
It likes us.
It does.
All right. Seen enough of that?
I've seen enough of that. That was cool. That was a good fish. Thanks for finding me a cool fish.
Wide view is giving us a really nice view of some of those yellow bolosomas.
Oh yeah, there's a whole bunch of them around
There's
here.
a whole bunch of them.
Your big cheeses.
The big cheese wheel moons.
If only they were cheese.
If only.
Megan, can you say the kind of fish it was again? I think a couple people just tuned in right at the end there.
So the name of this fish was Eridmictes.
Very pretty. Also, Val, welcome. We did introductions earlier. Do you want to introduce yourself real quick since this is the final dive and the final watch?
Sure thing. So, apologies for being late. I was up in the studio helping Matt with an interaction with some of his students back at his home institution. So my name is Val Finlayson. I'm the watch lead for 8 to 12, sitting in the Science 1 seat. I'm one of the science co-leads and the geologist on this expedition. The rest of the time when I'm not at sea, I work at the University of Maryland at College Park, where I am an isotope geochemist, and I do a lot of work understanding the origins and composition of the rocks that we pick up on ships like Nautilus. And I actually have some NA138 samples in the lab right now, waiting for me to work on them some more when I get back.
Was that your first Nautilus cruise?
That was my first Nautilus cruise.
Cool.
Yeah. Had a blast. Yeah, I have a little bit more data on some of those to collect, and then I can finish writing up the paper I have in progress on those. So that's pretty exciting.
Very exciting.
Yeah.
I was talking with Steve earlier about the timeline for identifying new species. He said it takes approximately, on average, 10 years between when the species sample is collected and when it's actually made official.
That makes me feel a little better about
yeah,
how long it takes to publish a geochemistry paper.
Mm-hmm.
That can, if you're lucky, it takes a year or two. It can usually take 3 up to sometimes 10 years.
Yeah.
Depending. Sometimes it's hard to get them published.
Why is that?
It depends a little bit on the nature of the topic and the peer reviewers that you get. Some people can be very, very critical, and I believe that is usually a good thing, but some people less so. Other times it's just really difficult, finicky work. So there's the component aside from just sometimes the time it takes in review. There's a component of having to spend a lot of time in the lab collecting the data too.
Oh.
So
no, it's just a bubble.
Yeah, a lot of it's purely science, but there is an element of getting it through the community standards of peer review and occasionally some politics with it, which is strange. You would think that there wouldn't be the politics problem in geochemistry, but there is, just like in any field. Yeah, I had one paper that came out a few months ago that had been in review for 3.5 years, which is ridiculous.
Yeah.
Yeah. Others I've had in review for about 3 months, and I've been slow to return revisions, and that's on me, but I've been trying to bank up a lot of data this year just so that it's there and available for me to work on, just in case. So I've actually had a little time to work on those revisions on this ship, and it's been really nice. Yeah,
oh, that's good.
Here and there. Not the last couple of days because we've been super busy, but you know, you take the opportunity when you can. Ten years though to get a new species described.
Yeah,
that is a very long time.
Most people don't start on it right after the collection.
Right.
You know, some of these animals that we collect are like, it's a new species, and you know, they go into the queue, sort of like some of the video. It goes into the queue, it doesn't get annotated immediately. Sometimes it does. Like, we've already completed annotation on the NA167 and 168 Palau dives from last year.
Okay.
Those are complete. But they were a priority for Palau and for some of our grad students.
Gotcha. Yeah, it's the same sort of thing with geochemistry, and you also have the added layer. I don't know if this is the same. It's probably the same.
Right? Yeah, so there's like a little hat on top.
A hat on the sponge.
Yeah.
Yeah,
I don't know if it's another crinoid or. Yeah, it's a crinoid.
Okay. Does funding also affect the queue?
It can affect the queue, yes.
Yeah, I have a bunch of 154 samples that I'm keen to work on, and I need to secure some funding for those.
We have completed 154.
Cool.
Yeah.
I have not.
Those are annotated.
That's awesome.
But that was up in the Papahanao Mokuakea.
Yeah.
So that's generally a priority for our funders at OER and NOAA and NOAA deep sea corals. So
makes sense.
We do a lot of those, and all the Okeanos dives in the Pacific we annotate.
Are we going?
We just completed a 30-meter move, and I was going to keep moving.
Yep.
Is it just me, or is that sponge really choked up with sediment?
It is. It's very dead.
It's dead. Yeah, it is very full of sediment from the way it looks.
I was kind of hoping that the little top hat was a ceryanthid, a tube worm, but it was a crinoid.
It's a crinoid.
I like crinoids too, but
they're cool. They just hang out.
Yeah, we see them a lot. And,
yeah.
A tube worm would have been cool. But you never know until you look. And that's the nice thing of actually being here in the moment. I can go, enhance, enhance. Instead of, you know, grumbling at my screen like, oh, I didn't look at it. See, now, if we don't look at it, it's totally my fault.
Do you ever have those moments when you're annotating footage that you've helped collect? You're just like, past Megan, why didn't you zoom?
Oh, absolutely, 100%.
There's stuff that we don't see while we're in the moment. It's easier to spot it after the fact.
Oh,
sometimes.
absolutely. Only when you can pause the video.
Yes.
And it's not like you know, I'm 100% glued to the main HD screen. I've got a lot of other screens to watch, and you know, people to confer with. And so it's a multitasking environment here.
Yeah, you're kind of the hub of the watch.
And you guys have a lot going back there with the sea log and the Multipuffer. So, you know, having all the eyes in the van watching at different points in time definitely helps.
Definitely, yeah.
I rely on our video person to have good eyes with their multiple screens over there in the corner. Sometimes other people spot things that I don't spot.
Yeah, exactly.
Like the tiny single polyp from last night. They're like, oh, there's a little floaty thing underneath. Like, what? I didn't see that.
Yeah, that was Amber who spotted that.
Yeah,
and,
it's got the good eyes.
Yeah, she's got a nice 4K screen over there too, so she can see some of those things the rest of us may not be able to.
If Megan had this screen, oh man, unstoppable.
I do have a 4K screen in my office that I use.
Nice.
But the video isn't 4K, so it doesn't really matter, but it definitely helps. There's a pneumatocarcinus up ahead. Oh, there's another floating little shrimp.
Yeah, let's take a look at our shrimp friend.
So this is an acanthophyra. Another way to tell is it has this little hump on the back. Oh, it's gone. You saw that on the tail. It looked like it had a little hunchback.
Mm-hmm.
That's because the pleons, so like the scales on the tail, the second one overlaps the first and the third, which gives it that sort of hump look. Whereas some of the aristeid shrimps and the other big shrimps that we see, the first and the second and the third are all just overlapping each other, one after another. And so it doesn't, those shrimps don't have that humpback.
Got that. So yeah, knowing those little morphological tricks is a great way to help differentiate between species, but not for everything. Doesn't work so well for bamboos.
No, no. Not even the samples that are in the lab.
Oh,
that
man.
you have a really close look at them, still you don't know what they are.
Sneaky genetics.
So sneaky.
Yep.
Yeah, because we used to be like, oh, if it's just nodally branching, it's a completely different than the ones that are internodally branching. And then we're like, we found one that does both.
Oh, no.
Okay. Now what is it? I don't know. And then you're like, oh, well, maybe it's, you know, this one has extra long intertentacular spines and this one doesn't. They must be like completely different.
And the corals are just like, nah, we're just going to keep throwing you for a loop. We're just going to do all the things.
And some species were described only based on material that was preserved for a very long time. So we don't know how they look underwater.
Yeah, because they change quite a bit once you get them back to the lab. Yeah.
The color changes because of the ethanol.
Yeah, yeah.
Wow.
And don't some species, not necessarily the corals, but don't some species, once they're preserved, they change size too?
Yeah, they can as well, because the tissue gets like dehydrated because of the ethanol, and they shrink a little bit.
Yeah, I remember Steve mentioning that with some octopus that he's worked with, some octopuses.
Yeah, and cephalopods get like stiff.
Oh, interesting.
Yeah. Oh. With time in ethanol, they get stiff. It's strange.
Does it just change something with their proteins or something?
I don't know.
Huh.
See what this little blob is?
Just a bump.
It's like an old holdfast, maybe?
Yeah.
Yeah.
Oh. It's a bump on a log in a hole in the bottom of the sea.
Literally. We're just missing the log part.
Right.
And,
oh man, if there was a log.
Steve's spider sense would start tingling.
Yeah, woodfalls are really,
Those
really
are fun.
wild.
Yes.
The way he talks about them, they're very special.
There's a lot of weird stuff on woodfalls.
Yeah.
There are these really crazy bivalves that look like worms. So they're called shipworms.
Oh, I've heard of those.
Yeah, but they're not actually worms. They're mollusks, which threw me because they don't look anything like mollusks. And then my coworker Sarah, she saw that I had annotated that and was like, "It says it's a mollusk, but it looks like a worm to me." I was like, "And you would be right. It does look like a worm, but it's not."
Very probably named for how they will colonize the hulls of ships. I remember reading about how that used to be an issue for some of the old wood-hulled ships way back in the day.
Yeah, the wood gets consumed. It's a good food source.
And the wood eventually gets a little more porous if you get enough of them, and that's not great if you want to be
Keep
floating.
the water on the outside of the boat?
Yes.
We do like having the water on the outside of the boat.
Very much so. All right, so we are currently right about on top of waypoint 13 and making our way up toward waypoint 14, which is the last waypoint we have planned for this dive. But
then we can go rogue.
We have, yeah, we have quite a bit of dive time, on bottom time left after this. So yes, we are gonna go rogue. Watch out.
Yeah, that's our last planned waypoint.
Wow.
We didn't think we were gonna make it this far, but I've said that every single.
Shallow.
We're only
well,
like
we started really deep.
we're less than halfway up this ridge
We
toward the
almost went
summit.
1,000 meters up.
Yeah.
Wow. Hercules getting his steps in this dive.
Yeah, getting the steps in.
I just imagined a Hercules with big muscles.
And that's why it's called Hercules. Yeah, I've said those words most dives this expedition, but we have been consistently making tracks, and I think it being a little bit less populous on these seamounts has a bit to do with that. So we're underestimating the ground that we're going to cover pretty consistently. There are other places where we dive where there's just so much going on and so much to see that we may not make the end of the track. And it's not like we're required to or anything. It's it's a little, you know, we don't know what we're going to meet down here. So there's always a little bit of careful estimations going into the dive planning that may or may not be very accurate.
Did they ever find their abyssopathes?
No.
I don't think so.
We're still looking for it.
Steve was saying it might be a little shallow.
Yeah, it's a bit shallow now.
Yeah.
Last night it was more in the range.
Okay. Yeah, he didn't seem too optimistic that we would find one, but we kept an eye out.
Is this the cheese whale?
Yeah.
Yeah. They're looking more cheesy.
They're getting a lot bigger.
Cheese whales.
It reminds me of what we saw up in the Liliuokalani Ridge up in the northern part of Papua New Makuakea. And from what I understand, they were quite large in the Voyager seamounts too, in the southern part of the monument.
Mm-hmm.
Oh.
Yeah, I've seen some really, really massive ones up there.
And you also get just these monster polyopagons too.
Oh yeah.
It's very cool. Monster
and
corals.
Don't forget about the minivan sponge.
Yeah. Those are fun. Yeah, I don't
That,
remember which
that sponge has a
seamount.
really nice, like, scratchy Christmas sweater texture to it. Yeah, had one collected, or a piece collected in 2017 on an Okeanos cruise, and had to deal with it. And it was such a weird texture.
Interesting.
Interesting.
Yeah, I wouldn't eat it, Levi.
I think
But
he's
you
still
could
thinking about
wear it for
breakfast.
Christmas.
It would be just as itchy, right?
Yeah, oh my God, it was definitely itchy. Probably more itchy.
I don't remember which seamount it was, but somewhere in the Liliocolanies, we did do this entire dive where it looked like the seamount was wearing a sponge sweater because it was so thickly coated in dead sponges for most of the dive track. It was incredible.
Yeah.
The infamous sponge dive.
I love that there's an infamous sponge dive.
Yeah, we annotated
There are
those
probably a
ones
couple
as well.
I'll bet you did. So you just get to hear us on those recordings just going, there's just so many sponges.
Yeah. Does a high density of sponges usually mean something, or they just happen to
Well, a
fall
high
there?
density of any animal means something.
Yeah.
That was a nematocarcinus that's drifting. You saw the shadow. It means that there's food.
Yeah.
If there's animals.
Oh.
What it means if you see a lot of sponges versus a lot of corals, I'm not so sure.
Yeah.
But that is a great question, and I would love to answer that someday.
Right. Yeah, there was also the same set of seamounts where we did another dive, same expedition, where we just saw acres and acres in some of these places of hemicoralia. They're just all over the place.
Oh yeah, the super dense hemicorallium fields.
Yeah.
Oh, they're so beautiful.
Those are so striking.
What do they look like?
It just looks like a garden of pink everywhere you can see. It's so dense that you can't put the vehicle down.
Oh my gosh.
Yeah, unless you really want to sit on a coral.
Oh.
And they're so fragile.
Yeah.
Oh.
So we flew over a lot of it.
Oh, well, that's good. Protecting it.
Yeah, those are one of those where your annotations are 100 plus, correct?
Yes,
yeah. It's way too much to count.
You're like, they were there.
Do you have a button for infinite?
Infinite animals.
Yes.
I just keep hitting the 100 plus more and more frequently.
Until something goes haywire with the program and you get an integer overflow and it's like,
oh no,
too many animals. The program must now shut down.
Usually I'll put a note like cannot count, impossible.
Yeah.
Wow.
Statistically, once you get over a certain number, it's not going to make as much a difference.
Right, yeah.
Oh.
But it is important to note when it's just an insanely dense population, because that's something you'd like to know.
It's not something you see every day.
Exactly.
Yeah.
Making notes of really unique occurrences is important.
Yes. Do you ever do like contour maps of like densities of species on the seafloor?
I have tried that with some of the lava flow coral garden work that I was doing, just to see how along the dive track the density changed, and sort of did a little bit of a heat map along the dive track, and that was kind of cool.
It is.
It's hard because we're just going in a straight line, so.
Yeah, it's hard to contour.
Yeah, exactly. You just have a little pencil scribble.
Is it possible to do any of that more regionally, or is it just too complex?
It's a bit complex.
Yeah.
And not enough data.
Gotcha.
If we did sort of like a grid survey on a seamount, then maybe we could, or like we kind of mow the lawn the way we do with mapping.
Mm-hmm.
We could
It'd
do
be a little
more
different.
of that.
Makes sense.
More transects.
Yeah. No, I ask because I do enjoy a good contour map.
Right? Yeah, the hard thing is we go to these places only once.
Yeah.
And we do one line and we move
that's
on.
it.
Um,
Yeah, and then when you do, say, 3 dives on King George Seamount out in the Liliocalanes, all 3 sides are totally different.
oh, absolutely.
Yeah. So how do you deal with trying to map that out?
And then, you know, our different sites are so far apart from each other that you can make a map, but it's like drawing dots on the world. It would be like, you know, you just took your major cities and like, these are our sample sites, like, and you're making assumptions about the entire country based on, you know, the 5 cities you visited.
And you know what? That's exactly what we're doing with the isotope work right now. It is not perfect. It's a known issue. Yeah, that's one of the interesting and frustrating things about my job is with these uncharacterized seamounts. Often I'm working on 1, maybe 2 samples to do the age and characterization, like source characterization of these.
Shrimp.
Yeah, big shrimp.
Nice big shrimp.
Chunky
So
shrimp.
this is the shrimp that I was telling you about, that the pleons, those plates on the tail, they only overlap in a row.
Oh.
So they tend to be the faster swimmers. They're always cruising. And they have larger pleopods compared to the Acanthophyra. And those are the swimming legs.
Yeah. Yeah, being able to do only 1 or 2 rocks per seamount, even for dredge cruises, you know, it's just what's in the budget and what you can reasonably handle as far as human hours, you know, the human labor aspect, especially for larger, more spread out areas like this. You know, the question is always, okay, we have a data point that gives us a general idea of the seamount. What else are we missing in there? Because, yeah, 1 sample, 2 samples can give us a pretty good idea of what the mantle componentry is that is building this up, and that does tend to be oddly consistent. Like, it works, but there's so much more nuance to the actual seamount building process that you can see in the geochemistry that we're just straight up missing.
Yeah, well, doesn't the geochemistry kind of change over time of the lifetime of the building of the lava?
And we've got that characterized relatively well for Hawaii. It's still not perfect because each volcano, as we found out, is a little bit different in its geochemical evolution. And the way that even Hawaiian-style volcanoes change over time, and you know their shifts in geochemistry doesn't reflect how a lot of these smaller seamounts geochemically evolve over time. So we've got like Hawaiian type volcanic stages, and then we have like the Louisville and the Easter type volcanic stages. So you know, you can usually predict based on a few physical factors which type you're likelier to be looking at, but we don't know that for sure because we're not able to characterize that entire stratigraphic section. And the best shot that we have of getting like the real like snapshot of the seamount geochemistry that we're after is why I'm always talking about wanting the shield stage samples. And that's the main volcanic building phase, the thing that really builds up the platform. That's where you're getting the most melting happening, the most melt production. It's the most representative of the source that melt is coming from. So that's why I'm always like, I love the deep dives. You know, that's where we get the good rocks, the most representative stuff. And we don't always know in the shallower areas, you know, are we moving out of that shield stage and into a much later stage where that geochemistry does start to shift a little bit? And if so, how exactly is it shifting? You know, is it doing the Hawaiian thing? Is it doing the Louisville or Easter thing? We don't entirely know. And it leaves a lot of open questions, but it gets us at least in the ballpark.
Yeah, that makes sense.
Yeah, yeah.
Was there anything that we saw here on this expedition that you didn't expect geologically?
Geologically?
Yeah, any surprises?
Yeah, we saw that a couple of dives ago, that seamount with the weird tectonic patterning in it.
Yeah.
And I was kind of hoping to see another one of those after we documented 2 or 3 of those last summer in a different part of the ocean. But I wasn't expecting that we would find it. You know, it was just more, hey, it would be cool if we saw this.
Yeah.
And we got lucky and we did.
Yeah.
And yeah, it was weird. Which is a good thing.
Yeah. Has more questions to solve.
Now, what it was doing there is another question. That's an open-ended thing. That's something that some of my teammates and I we're trying to figure out what is causing that kind of seamount to form, because we don't understand it.
That's a big undertaking.
We sure. I mean, that's the job that we do. That's the thing that we choose to do. And
yeah.
That's the thing that I like about science, and you've heard me say it more than once. If it were easy, it would have been done already.
Yeah, it's true.
And we are kind of doing things in hard mode geologically with these seamounts. They're not easy to sample. You don't necessarily know with 100% certainty what kind of rock you're picking up and what it's going to be suitable for analytically. We're mapping volcanic structures that were previously blobs predicted by gravity anomalies measured by space shuttles. And we're actually going and ground truthing this for the first time. So everything is a little bit of a surprise here.
Yeah.
Yep.
It's wonderful.
Finally found the up.
Okay.
Alrighty, I'd like to give a shout out to those of y'all just tuning in right now. This is our last dive of the NA174 expedition. We are currently on a seamount south of Bikar in the, seamount south of Bikar, and we're exploring. I would like to give a shout out to everybody we have a lot of viewers from all over today again.
And what's what?
What's what?
It's a fish.
Oh. Ooh.
It's fast.
Too fast.
It does not want to be perceived.
Oh. Or maybe it does. No, it doesn't. Okay. Never mind. It took another turn.
Yeah, so that one looked more like Bazazitas for the little bit I could see of the pectoral fins.
It looks like it had a pretty rounded head, so it didn't look like an eel at all.
Yeah, no, it was definitely a cuskeel.
Oh, it was?
Yeah.
Okay.
Oh.
Yeah, 100% a cuskeel. But remember we saw that erycmyctes, the erycmyctes and the Bazazitas look very similar.
Okay.
And they can co-occur.
Gotcha.
So it's hard to tell which one is which. And the main character is that pectoral fin. The Bazazitas doesn't have that two-lobe bit. The males are obviously very... can we look at
that? Was a weird shape for that rock.
rock.
Can we take a closer look at that? That was
Levi's?
odd.
Yes.
Do we have...
What are we looking at?
It's just below
you.
the field of view.
Okay. Oh.
It It looks like a narwhal tusk, except
Oh.
It could be a bone.
That's what I was wondering too, because rocks don't do this.
Oh.
So you see this right
what
here?
Is that?
Yeah, it could be like a whale bone or something.
Yeah, this morphology is not normal for this kind of rock.
Whoa.
Which is making me also think it's biological.
Oh, wow.
Yeah, sorry, I know this is really inconvenient. Yeah. No, I understand. No, we got some footage of that. That we can take a closer look later. Thanks.
And because of the manganese crust, that probably most likely be a fossil, right? Or
I didn't quite catch your question there. You're a little quiet in my ear.
Oh, no worries. So for the crust on it,
nope,
would it be
we are not moving.
It'd be a fossil or just bone under there, or how does that preserve it?
I don't really know the true demarcation between bone and fossil, but it's encrusted, so it's quite old.
Oh.
Yeah. Yeah, that's a substantial wall. We'll keep an eye out for more bones, or bone candidates. Let's be careful there.
Yeah, I couldn't say for sure.
Yeah.
It could have been a really weird rock. You never know.
Rocks do not break like that. Like that would be
yeah,
really,
no, that
really
definitely didn't look like a rock.
Yeah.
But, you know, sometimes we're like, oh, it's a bone, but it's actually just a sponge.
Yeah. Well, I did see a patch of sediment that looked like a sea star already, but it was very clearly sediment.
Yeah. There are a lot of star-shaped sediment patches.
There are. And they give me so frequently.
Is there a reason we don't sample possible fossil candidates or bone-shaped candidates?
Oh, we do. We do sample those things.
Oh,
we just have to keep the vehicles moving.
Yeah, it just was bad timing.
Oh, dang.
Yeah.
Atalanta happened to just be swinging at that moment in time when we came upon, and we hadn't been stretched out yet, so we didn't have a lot of layback time.
Yeah.
In that particular moment.
Yeah.
There are, depending on where you're working, like if there are certain, like in Papahanao, Mokuakea, there were some places where we did see a lot of fossilized whale bones, but we would not have been able to sample them because that was a material that was specifically something we were not allowed to sample as laid out in our permit.
Oh.
So there are places where, yeah, we're not allowed, but then there are other places where, you know, if the opportunity presents, we may choose to take a sample.
Yeah.
Yeah, there are some very strict sampling guidelines and permits for the Papahanao Mokuakea.
Yeah.
And there's also strict guidelines about what we can and cannot leave behind if we need to drop weight in order to recover the vehicle.
Oh.
So we had to have a number of meetings about that before we were able to go up there with Lu'ukai, because we use a different ballasting system than we do on Hercules.
Mm-hmm.
And then I guess for those watching, why is it not so simple to just backtrack and turn Hercules around?
Well, to go back, we would have to back the ship up, and then we would have to back Hercules up, and Hercules doesn't like to go downhill. So it takes a lot of time to go backwards.
It's logistically very challenging, yeah.
We can do it.
We've done
in the event that it's like, this is really, really important, we'll go back, but that costs a lot of time. Yeah. And so it's a balance. We want to balance how much distance we make and headway we make on our track, and with the importance of the sample that we want to collect if we passed over it.
Yeah.
All right, so we are hearing from Jonathan Womack in the shore team chat, and he's suggesting that what we saw there had the right shape for a dolphin skull or something similar.
Okay.
Oh,
yeah.
wow.
So he's sending over some reference pictures now. I'm just waiting for it to load. Oh, that's a nice screen grab.
Yeah, it's kind of rare to find bones in the deep sea because there are animals that actually consume that material.
Oh, wow.
Ready for a move?
Oh, yeah. No. Yeah, I'm looking at some dolphin skull pictures here. Yeah, that does have a lot of morphological similarities.
Mm-hmm. Yeah.
Oh, that's a good angle. Thank you, Jonathan. Oh, yeah, yep. I can see that.
Let's see. I want to look at your computer. You
I don't have it up on Chief's side, but we have it up on Science 2. Yeah, sorry, I'm looking on my laptop.
Okay.
I would let you KVM in if it were connected.
Oh, yeah. No, I'd agree. That looks the same to me. That's pretty cool.
Yeah, yeah. This is why we have our shore team. They are an essential part of helping identify some things that we don't have the expertise on the ship. Yeah, yeah.
It was a cetacean.
No, it's so helpful just to have extra eyes on shore.
Well, and they can pause.
Yes. Yeah, we can't.
They can scrub back, you know.
It's a little harder for us to do that.
These are more cheese wheels. Oh, they're lined up.
Yeah, I think that's going to be one of the things I look forward to when I get home is some cheese.
Yes, me too.
I agree.
We've been having this like it's the shape of like American cheese slices, but it's very different flavor.
I've
color.
Not tried it.
Yeah, it was different.
Oh.
I couldn't quite put my finger on what kind of cheese it tasted like. It tasted like 3 different kinds of cheese all at once.
Huh.
Oh.
But it didn't taste anything like American cheese, but it had the texture of like a Kraft American cheese.
Interesting.
Oh.
Yeah.
Yeah.
But it was like a little bit of a white cheddar, kind of a little bit of a Swiss flavor.
Huh.
That sounds pretty good.
Yeah, I think it would be best melted. Like
Probably,
if you did like
yeah.
a panini situation. Grilled cheese. Oh, I could go for a grilled cheese.
Ooh, yeah.
I thought we had the sandwich press thing up there still. Did they move that?
Yeah. It keeps coming and going. Like it was there and then it was gone.
A phantom.
Well, when we leave from Honolulu, sometimes I bring my own cheese.
Ooh.
Mm-hmm.
Oh yeah, yeah, Danny got some Cheetos from the bulk shop, and I think they were like Australian or New Zealand Cheetos, and they have actual real cheese instead of, you know, the neon orange flavor powder that we're used to.
Yeah.
How was the flavor of the real cheese Cheetos versus the not real cheese Cheetos?
I have a bag of those up for grabs, actually, because I had them stashed away, and I realized they have gluten in them.
Oh, no.
Oh, what they do?
No, that's okay.
No.
It's actually a really common thing I found when traveling